viendo un proyecto juntos, él me pidió pues, si le podíamos hacer las neuronas para venir aquí.
¿Y qué es lo que pasa ahí dentro?
Si son Alzheimer, hay neuronitas que son un poquito punchas.
Las neuronas epilépticas lo que hacen es que se ven como así.
Sí, exacto, para que los organoides sean medibles y predecibles.
Que además habéis subido del 94 al 95% este año. ¿Cómo has hecho esto, Oscar?
Con magia, todo es magia.
So imagine if we scale it up to more higher number of neuron cells, what other stuff we can do. It is kind of sci-fi, but at the same time it's not, because if you're thinking about your own brain, that's how we learn.
¿Sabías que en Mobile World Congress 25 se presentó el primer ordenador biológico del mundo con neuronas reales que viven en un mundo digital? ¡Boom! Sí, como lo oyes. Y te preguntarás, ¿qué narices es un ordenador biológico? ¿Neuronas? ¿Cómo? ¿Para qué? ¿Por qué? ¿Esto qué es? ¿Matrix? Pues un poquito, ahora entiendes lo de las gafas, ¿no? Y espero que también el guiño de la tecnoperra fucsia. Quédate conmigo y tendrás tus respuestas, porque el capítulo de hoy es un episodio especial para Cortical Labs. Bueno, combinación, Cortical Labs y organoides. Lo descubrirás ahora. Ah, por cierto, pedimos perdón porque tuvimos un problema de sonido. Había un dispositivo por ahí que no logramos encontrar, que producía un montón de interferencias. ¿Será la Matrix? Estás entrando en Rivela, universo TPG, donde descubrirás tecnología que rompe esquemas, cambia industrias y define el futuro del mundo. ¿Ready para hackear el presente y construir el mañana? Sigue a la tecnoperra fucsia. A ladrar. Wof, wof.
Estamos aquí con una de las protagonistas de la primera temporada, la doctora Sandra Acosta. Sandra, ¿qué tal? ¿Cómo estás?
Un poco overwhelmed con tantas luces.
Sí, es un poco agobiante, ¿no? Esto es el paraíso de las luces, se pasa un poco.
Sí, un poco preepiléptico.
Bueno, Sandra, vamos a ver, hagamos un recap. ¿Qué es lo que habéis traído a MWC 25?
Hemos traído organoides directamente, los tenemos aquí y los estamos viendo cómo los analizamos en pantalla. Sí.
Cuéntale un poco más en la cámara. Venga.
Pues estos son organoides, justo los que estamos viendo aquí, los hemos marcado. Estos organoides vienen de pacientes que tienen Alzheimer, los hemos crecido durante 6 meses en el laboratorio y los hemos teñido para poder ver las células. Y aquí los tenéis puestos con nuestra colaboración con Cortical Labs encima de los electrodos, que es como mediríamos la actividad eléctrica de estas neuronas.
Un organoide es una estructura tridimensional en miniatura que imita la arquitectura y algunas funciones de un órgano real. Estos órganos en miniatura se crean a partir de células madres. Si quieres saber más, pincha en la tarjeta para ver el episodio de la primera temporada donde Sandra nos explica esto y muchísimo más sobre su proyecto.
Yo tengo que preguntar 2 cosas. La primera va a ser muy tonta, pero ¿los habéis tenido de rosa? De lila. Oh, no pasa nada, te sigo queriendo.
Hay algo de rosa, pero
no
más
nada.
lila que rosa.
¿Les habéis puesto electrodos y por qué?
Los electrodos hemos puesto los organoides dentro de la máquina de Cortical Labs para medir la actividad eléctrica, porque las neuronas son electricidad.
Bien, entonces, a ver, vamos a ver el dispositivo, ¿vale?
Tenemos los organoides creciendo aquí, los ponemos y aquí hay unos electrodos que mediante todo el flujo de medios de cultivos, aquí se hacen los recordings, lo que llamamos, para medir la actividad eléctrica de las neuronas. Entonces, cuando aquí pones un estímulo concreto, por ejemplo, se le pone un fármaco o algo, pues podemos ver cómo se modula esa actividad en las neuronas que están siendo tratadas en el incubador dedicado de Cortical Labs. Y nosotros tenemos aquí pues unos organoides. Estas bolitas son los organoides de cerebro que crecemos en el laboratorio. Estos tienen aproximadamente 3 meses de crecimiento. Los hemos obtenido de células de la piel de pacientes y les hemos puesto la señalización para conseguir que sean neuronas y al final se convierten en una bola de neuronas y otros tipos de células y que tenemos en el laboratorio. Y luego hacemos todas las otras pruebas, los ponemos en la máquina, les hacemos los marcajes que necesitamos y bueno, analizamos lo que pasa ahí dentro.
¿Y qué es lo que pasa ahí dentro?
Pues si son Alzheimer, pues que hay neuronitas
que están pochas.
Son un poquito ponchas.
Ya,
sí.
No pasa nada.
Y si son epilépticas, pues que están un poco hiperactivadas.
¿Sí? O sea, las neuronas epilépticas lo que hacen es que se ven como así.
Sí, exacto. Una crisis epiléptica es una hiperexcitación neuronal. Entonces puede ser o porque las neuronas excitatorias están hiperactivadas o porque las inhibitorias, que son como el freno de esa actividad, no funcionan correctamente. Entonces tienes que saber muy bien qué tipo de epilepsia es para poder dar el tratamiento completo, que esto es lo que nosotros hacemos en Organoid. Buscamos el tratamiento concreto para esas células porque ya las tenemos, sabemos cómo está su actividad.
Y porque esto es muy relevante porque es mucho mejor hacer tratamientos con organoides que con personitas. ¿Cuánta era la efectividad del algoritmo?
Muy alta, 0,95. Y además tienes la suerte que el algoritmo lo hizo Oscar.
Hola, Oscar, ¿qué tal? Yo soy
Mi
Silvia.
colega, nos conocemos desde hace 7 años cuando aterricé en Barcelona y él ha sido el artífice de este algoritmo que hemos estado trabajando en los últimos años y que ha puesto a punto para que los organoides sean medibles y predecibles.
Y que además habéis subido del 94 al 95% este año, o sea que enhorabuena, Oscar.
Gracias, muchas gracias.
¿Cómo has hecho esto, Oscar?
Con magia, todo es magia. No, con mucha prueba y error y luego pues teniendo mucho conocimiento de los algoritmos, ¿no? Entonces prácticamente hemos hecho, muchas de las cosas que hemos hecho se llaman transfer learning, que es coger partes de algoritmos que ya se han usado para otros procesos y los hemos adaptado para los organoides.
Claro, esto es continuous improvement.
Sí,
Estamos mejorando, trabajando para mejorar
sí.
cada vez más.
Evidentemente las arquitecturas de lo que serían los deep learnings son dependientes de problemas. Y entonces eso quiere decir que a pesar de que hemos usado transfer learning, es posible que existan otras arquitecturas que sean más eficientes desde el punto de vista tanto de consumo de recursos como de producción de resultados que todavía están por descubrir. Eso es lo que estamos también trabajando.
Qué interesante. O sea, aquí desde el desarrollo, claro, tú me habías contado esto, pero no me habían contado la parte técnica.
Bueno,
Cada uno tiene que hablar de lo suyo, ¿no?
Esa es la clave, ¿no? Montar equipos con gente que es maravillosa en lo suyo y que luego pues van como una bomba, como vosotros. Y además ahora colaborando con Australia.
Bueno, sí, la verdad es que sí, nos ha venido, conocimos a FON el año pasado, gracias a ti. Y
Gracias.
Gracias a Silvia,
Tranquila.
Silvia Rivela, que es la gran nexo de unión. Y desde que conocimos a FON el año pasado, nos hemos visto en 4 o 5 sitios, estamos escribiendo un proyecto juntos. Él me pidió pues, si le podíamos hacer las neuronas para venir aquí.
Han just told me to come.
I didn't know they recorded. And Azin,
Hello, Hon.
Azin has been collecting the neurons from my lab to bring them here,
Yeah.
and they are in those incubators.
They told me that on Sunday they were...
We spent 8 hours in the lab. Yes.
Look at it, she's the butcher. She's the one who's actually cutting
Man, I didn't cut anything. I just tried to maintain them for long term. So
Put them there.
actually I'm the angel.
You're the angel, you're the saver.
Not the same.
Saver. Exactly, it's the saver. But we worked very hard on Sunday and yesterday to get some neurons here.
Yeah.
What
So
is
we're
the-
working on Sunday weekend, hair weekend. Yeah.
What is the hardest part to actually be able to put those neurons here?
Ooh, there's many hard parts, right? So it can face
You
so
mean-
many different times, so many different procedures that can be harmful. So we've been working for many different things. So besides of like the growing process, the maturation process, they require the right time. So that it needs to be very controlled. But my lab is like 1 kilometer down the city from here. But we had to walk through the 1 kilometer inside here with the whole system in hands. So she was walking, so that's terrible for a neuron.
For the neurons. And also for you.
For the arms too.
Ay, yo no puedo evitarlo, pero a mí me dices esto y esto me recuerda a la comunidad del anillo. Hay 1 kilómetro caminando con las neuronitas. Tal cual.
So what we did was that we got like a transportable battery because that needs to be pumped. It's like a human body. You need to continuously have a fluid flow on top of the neural cells sitting on that. So we got one of that batteries connecting to that. And while the battery was connected, we just walking around just because we need that to flow, right? But other than that, it was. So as long as we have these cells on the chip, we can easily transport it and put it on the top of that. And then the rest is filling the bottles with the media, which is kind of a food for the cells who sit there. And then the rest is using that peristaltic pump to pump the media on top of the cells. And then the cells sit there. Then we can connect it here.
So you were saying, what is that?
So this is the CL1 perfusion system where the cells can sit in each of these. We have 30 of these at the moment here. They are connected to the CO2, which is very essential for the neuron cells to stay for long term. We have the control over temperature. Again, it's very essential to keep the body temperature, which is 37 degrees for the neuron cells. Otherwise, a small fluctuation on the temperature is going to kill the neuron cells. So everything have to keep nice and content for the neuron cells living in these CL1s. So 3 of these CL1s include Sandra cells, which she created in her lab. So we put it on these CL1s, and then basically we try to keep these neuron cells sitting in that for the long term. We have 30 of them, but the idea is having as much as possible of these CL1s, and of course as much as possible of neuron cells into these CL1s, so that we can do so much stuff like computing or even sharing our cloud space in our lab and doing so many cool stuff, or personalized medicine, which is another angle of the
Because we
research.
didn't say, but what is that?
This is the first biological computer that we can do computing stuff using the neuron cells living in this device. So the reason we need these CL1s is that when you culture the neuron cells and when you want to train them to do some specific task, you need a way of keep them for as long as possible, and this is the responsibility of CL1s.
You said task? Like what?
Like, for example, what we show is playing PONG. We show that by stimulating neuron cells in a specific way, they could learn to play PONG in the virtual environment. So what we did was sending some signal to the neuron cells, teaching them where the virtual ball is, where the virtual paddle is, and then by time they learn to respond to that signals we sending to them to move the paddle in responsible to where the ball is.
On their own.
Yeah.
Like you just, yay, do something, and they learn.
And this is the simplest task, I mean the initial task that we show that neuron cells are able to do it, but we have a long way of doing different sort of stuff and then teaching them, because this is just happening by writing a Python code and then sending signals to them, and then they learn to do some specific task. It is not something, it is kind of sci-fi, but at the same time it's not, because if you're thinking about your own brain, that's how we learn, right? We just learn by time that this is the way that you should function and this and that, and then we just have 100K of neuron cells on each of these chips. So imagine if we like scale it up to more like higher number of neuron cells, what other stuff we can do. But that 300K neuron cells was enough for us to understand the neuron cell function. And then of course we can go ahead and then increase the number of cells from 2D to 3D, and even increase the number of cells, and then they can do in parallel together, learn to do tasks in parallel.
This is a neuron army.
Yeah. Something like that.
This is a neuron army working for companies to actually process data to do good things.
Yeah.
Like treatments.
Yeah.
It's fucking amazing.
Another aspect that we are doing at the moment and we're focusing is personalized medicine, so we get the blood from patients suffering from, let's say, epilepsy, and then we convert it back to stem cells and stem cells to neuron cells, and then we put the neuron cells here, and then we can investigate different drugs while they're sitting here, they're living here basically, right? Because I told you it's like we have heart, we have like all the oxygen and CO2 coming to that, so they're living in this virtual body, and then we can inject some different drugs based on the, like, let's say epilepsy drugs, and it's kind of showing that, okay, for my cell line, this drug works better than that drug. And it's very interesting because you can, in that way, with personalized medicine, you can decrease the side effects that all of us can get from like generic medicine that are on the counter, and you can get it. So it's an amazing, another pathway that we all looking forward to that, yeah.
I can totally see that you love what you do.
Yeah. I really love it. It's very amazing because I love to do challenging stuff, and at the same time not doing the same time over and over. So I love it that like every time we discover new thing, and then it's amazing. Yeah.
What was that moment that you said, "Oh, this is fucking real"?
For me, was when we designing that PDMS structure, the octagon structure that was in the first.
Those one, the bodies. No.
Yeah, bodies, yes, but bodies was last year. The film that I can show you, the octagon films.
Let's go and see it.
Could become tumorous. They could be cancerous because it's the same cells, same genotype. So our immune system cannot differentiate between them and an infection.
Tú, que aquí falta una pregunta clave. ¿De quién son las neuronas con las que trabajan? ¿Del equipo? La verdad es que se la hice, lo que pasa es que no se quedó grabado. Y nos explican que no quieren correr el riesgo de que estas neuronas entren en contacto con personas que son su origen. ¿Por qué? Porque resulta que si eso pasa, corren el riesgo de que... ¿Cómo te quedas? Yo me quedé loca.
Okay.
And so we don't know. It's a very, very tiny, almost in like very remote risk, but we're not taking any chances.
Okay, that's super interesting. So
Yeah.
you can actually manipulate your own cells
Yes.
because there is risky of cancer
Correct.
if you get in contact with them.
Yeah.
And you don't actually know. Well, not well.
So this is the reason why stem cell regenerative therapy hasn't actually really taken off yet, because there's still things we don't know about the stem cells. Right? Will they revert to become immature cell that can then replicate? Because if you want neurons, right, the neurons should not grow once they have them, because you only have very limited space in your brain, in your skull. So this is the reason why we haven't given humans stem cells back after a stroke, but the holy grail of regenerative therapy is if you have a stroke, we replace the neurons, so you go back to 100% capability. You have a heart attack, we replace the heart cells. Yeah, so we can have longer, more healthier lives.
Fun. Well, please
Yes.
introduce yourself
Yes.
because I didn't.
Yeah, so I'm the CEO and founder of Cortical Labs.
He's the one who was so crazy to actually create this first biological computer. When did you start it?
What did I study? I studied medicine at the University of Melbourne
And the company was created when?
in 2019, but I graduated in 2012.
Okay.
Yeah, so that was a long while ago. Okay.
Why did you start that?
I actually really wanted to become a doctor. I liked the intellectual challenge. I liked helping people, and I got the grades to do medicine. So I got into medicine, studied for 6 years as an undergraduate medical student, spent a year in the US doing research at Johns Hopkins University, where I was introduced to entrepreneurship there. And my final year of medical school, I did a project that somehow got funded by Microsoft for a grant.
Somehow?
Yeah, well, I guess it was quite lucky for us, and we did a bit of work around that. And that's how my first startup came about when I was in the second year residency. The device that we had built, a digital stethoscope that you could plug into a smartphone, a lot of parents were asking us, where do I buy this device and how much does it cost? And I said, hey, wait a minute, that sounds like a product. Maybe we should pursue this as a startup. So I convinced the mate from medical school to quit his job as a management consultant in Bain. He didn't lose anything. Nobody really likes working at Bain anyway. And joined me to create a startup. And we had a startup, we made these devices, and that was like how I got started going down this route.
Are you aware that maybe we are in the beginning of a new era of computing?
I think so, I really hope so. This is what gets me up and going every day, right? It's the idea that you don't wake up every day having created a new field of science, a new research field. And this is very exciting, and I want to share it with people. I want people to use this to do new therapies, right? Like what Sandra's doing to come up with dementia cures and therapies. I want people to use it to do new artificial intelligence. So I'm very excited about what we do, and this keeps me going every day.
How many people are in the team?
There's about 21 of us now.
21?
Yes.
So you grow a lot.
Yes, we have grown a lot.
I love it.
Yeah.
Well, thank you so much, Hon.
Thank you.
Let's go.
Yeah.
Let's continue to actually do the tour.
Yes. Okay.
Amazing.
Yeah. Thanks, Celia.
Si has llegado hasta aquí y te gusta lo que ves, suscríbete y comparte. Ayúdanos a seguir creciendo.
You asked me about what was the most interesting part of my last year. So it was when I designed this structure and we put it on the multi-electrode arrays and we inject neuron cells in a specific spatial coordination. So we could control where the cells are located inside the wells and where not located outside. And then we could kind of control how the neuron cells find each other and then spark together. So that was the amazing part. And when we find out that actually neuron cells, when they're sitting on this scaffold structure, they spark much better, they find each other much better. And then as a result, when we have more spiking, we can train them better. So that was the aha moment for me, an amazing part.
So basically, you are telling me that you created like a home, like a a comfy sofa.
Yeah, like a comfy sofa that kind of tell them, okay, connect to in this, sit in this well and connect through this pathway together, and now fire and connect together, wire together and fire together in this pathway. And then that was very amazing part of our project last year, yeah, end of last year. Yeah.
Tío, esto no es acojonante. Estoy flipando un montón. I'm like freaking out.
This is very amazing for me. And then, but at the end of the day, this is our main product gonna be CL1, where cells sitting there, and we wanna prolong leaving the cells in that system for as much as possible, because the longer we can keep them, the longer we can train them for the more complex task, right? So, and that is the...
And how long can you keep it?
We could keep it for a couple of months, up to 3 months at the moment, but our plan is focusing on like making that much more efficient, so that the longer, the better for us, yeah.
Well, actually, last year, last year, Hon explained to me that at the very beginning was 20 minutes. And afterwards, you created that device, not that, the previous version. It was 2 months, and now it's 3 months.
Yeah, well, it stays longer than that, but the activity, it shows us the activity up to 3-4 months, yeah. So the cells were sitting there, but they didn't show us activity. That's why we're thinking about how to make the situation better for neuron cells, and this structure help us to keep the neuron cells for longer, 6 months, they firing. And then now the second stage is putting this one with this scaffold into the CL1 machine and then see what's happening here.
When are you going to do that?
So as soon as I go back to Melbourne, this is actually on the go with different pattern, but when I'm going back, we're gonna focus on that side, because we've been very busy for preparation for MWC. But once I get back, I'm gonna start immediately on that and then see what's happening. Maybe next year we come with much more cooler stuff.
I'm gonna be here next year, but I hope I receive more news about that.
You're gonna get the news sooner than that. Yeah.
Please.
And this is part of the computing function that we are focusing on. We know that the fraction of learning is much faster than the AI model, but it depends on what sort of AI model. So in terms of energy consumption, we find out that neuron cells need much less, a fraction less energy of the AI models. And it's just quite natural, because if you think about human body or any live creature body, it doesn't need that much energy to function, for the brain to function. And then basically we harnessing this aspect of biology, less energy, but doing more stuff. And then that's what our culture doing at the moment.
One big learning that you had this year?
My big learning that is that just trust biology and trust nature, yeah. And you do the rest. If you know how the nature works and how biology works, you can combine it with engineering and you can do amazing stuff, yeah.
What's your background?
So my background is biomaterial and chemical engineering, and then I know cell culturing, I know engineering, and then I'm here at Cortical Labs because it's a multidisciplinary company. We have hardware engineers, software engineers, biologists, neuroscientists, and bioengineers like me, which I can describe myself as a bridge between pure engineers and pure biologists, so that I understand the language of both sides and I can communicate between them. And it's the amazing part of my job that I can be between both of them and I understand both of them. And it's quite cool, because you can be in engineering side, in biology side, and understand both of them and then talk to them, and it's amazing here.
That's super cool.
Yeah.
What was about your PhD?
So my PhD was a simulation of blood filtration in kidney, and that's actually that's the reason I came to Cortical Labs, because that CL1 has a filtration system, and then they just basically needs me to help them for this part of filtration system for CL1s. My PhD was computational modeling how the blood filtration happening in our kidney. Yeah.
I do love that, but I feel so silly sometimes. It's like, okay, I'm just an architect, and someone who's actually doing questions in a really not shy way. Something that you will say to the people, to the women
Don't scare if you see an area like programming, coding, or whatever, or math or science is dominated by men, because we women suppress for so long, so it takes time for us to go back to the scene. But we are capable of everything, and just trust it. Yeah.
And you are changing the world right now. So you dream big, you be inspired.
And then people are very helpful. When you're passionate and do your job properly, people want you. It doesn't matter what's your gender, actually, because as long as you do a good job, people just expect you to do your job properly, and then that's the moment that you can find your way and then go further and then further and further. Yeah.
The best message to actually finish that interview. Thank you so much.
So much for your time. That was amazing chatting with you. And thank you.
Jo, pues hemos llegado al final y yo voy a hacer un resumen. CL1 es el cuerpo que mantiene vivas a las neuronas. CL2 es el super rack con capacidad para 30 CL1s. Este sistema se podrá comprar y tener tu propio servidor de neuronas en tu laboratorio o donde quiera que tengas tu guarida secreta para salvar el mundo. Pero si todavía no tienes laboratorio, también podrás conectarte a su cloud, porque van a sacar un servicio cloud. Sí, como lo oyes. A sus servidores, conectándote de manera remota para utilizar sus propias neuronas a distancia. Yo no me he encontrado nada igual nunca. La verdad es que esto es tan loco que abre un tema ético bestial. ¿Qué sucede con estas neuronas? ¿Es que están vivas? ¿Dentro de 100 años podremos tener un cerebro real que piense de manera autónoma? ¿Qué opinas? ¿Nos gusta o nos disgusta? Déjanos tu comentario.